Thank you for the introduction. So this afternoon, my talk will focus on current product-specific guidances and common questions in pre-ANDA communications when developing orally inhaled and nasal drug products. So first, I will provide an overview on oral inhalation and nasal products. Then I will present the current bioequivalence recommendations for this type of products. Then I will turn the focus of the talk on the current product-specific guidances we have available. And at the end, I will present some typical questions we receive from generic industry when they are developing this type of complex products. Most of orally inhaled and nasal drug products are used to treat diseases in the respiratory tract. They are classified as drug-device combination products, meaning they have the drug component and the device component. And according to the GDUFA 2 commitment letter, they are also classified as complex products. They may have complex formulations, or complex routes of delivery, or complex dosage forms. And in many cases, all these complexities together in one single product. So the drug can be formulated as solution, or suspension, or a solid blend. The site of action can be systemic or local. There are different dosage forms according to nasal or inhalation routes of administration. So we have, for example, nasal spray products and nasal aerosol products. We also have inhalation sprays and inhalation aerosols. And the same for powder formulations for nasal or inhalation administration. There are multiple challenges that need to be considered when developing generics for local action, oral inhalation and nasal drug products. So first, the performance of the product depends on the device characteristics. Then, bioavailability of the drug depends on multiple factors. For example, how the patient uses the device to administer the medication. And when the drug is in the local site of action, all the process that happen, dissolution, permeability, clearance process. In addition to that, at this present moment, there is no practical way to measure the amount of drug that is delivered to the lung or nasal cavity. In the case of drugs that act systemically, they are delivered to the blood and then distributed to the sites of action in the body. In this case, bioequivalence can be determined with typical PK studies. For locally acting drugs, PK is downstream to the therapeutic effect. So in this case, a typical PK study may not be sufficient to establish bioequivalence. Currently, the agency recommends the weight of evidence approach for establishing bioequivalence of orally inhalation and nasal drug products. According to this approach, the generic product needs to demonstrate equivalence to the reference product in terms of in vitro studies, PK studies, a comparative clinical endpoint or pharmacodynamic study, in addition to formulation and device similarities, meaning all these elements together, A plus B plus C plus D. This is a list of in vitro bioequivalence studies we recommend for dry powder inhalers, metered dose inhalers, and nasal suspension products. I will not go over into detail of all these in vitro studies in terms of design and bioequivalence criteria due to the time limit we have at this presentation. These are the current in vivo studies we recommend for this type of product. So a comparative pharmacokinetic study for DPIs, MDIs, and nasal suspensions using healthy subjects. This is a typical single-dose, two-way crossover study. A comparative clinical endpoint or pharmacodynamic study for MDIs and DPIs using patients with asthma or COPD, depending on the indication in the RRD label. In the case of nasal suspensions, we recommend a comparative clinical endpoint study using patients with allergic rhinitis. For nasal solution products, the bioequivalence recommendation depends on the activity, if the drug will act systemically or local, and the formulation characteristics of the generic product. So if the product is intended for systemic action and the generic formulation is considered the same as reference formulation, in this case, in vitro studies only are sufficient. But if the formulation is considered not Q1, Q2, different from the reference formulation, then we recommend an in vivo PK study. For nasal solution products of local activity, we recommend sameness in terms of formulation and in vitro studies only. As Chris Andre presented you yesterday, these are the 4 ways of communication between generic industry and the agency. I will focus now on product-specific guidances. So we publish product-specific guidances with specific objectives to facilitate generic drug product availability in the market, to assist generic industry when developing these complex products, and to identify the current thinking methodology we believe it's most appropriate to support generic application. The bioequivalence recommendations in PSGs are guided based on the principles in the CFR 320/24, which lists different types of ways to measure bioavailability or establish bioequivalence. So considering all MDIs and DPI products and also nasal products we have in the market, currently we have about 55 and 60% of product-specific guidances for these products. This is a complete list of all PSGs we have available for MDIs, DPIs, nasal solution products, as well as nasal suspension products. You can go to the website and download all these product-specific guidances. Now I will focus my presentation on typical questions we receive from generic industry when they are developing oral inhalation and nasal drug products. So basically the questions are related to formulation development, device development, and bioequivalence. In terms of specific questions, sometimes asking clarifications on the product-specific guidances, or sometimes proposing alternative approaches to our current recommendations. In terms of formulation, this is the most common question we receive. If the proposed formulation is Q1, Q2 the same as the reference formulation. So the way we assess this type of question, we look into the excipients in the generic proposed formulation, if they are the same as the reference formulation, and also the concentrations of excipients, if they are similar to the reference formulation, within the plus minus 5% range. So when submitting formulation-related questions, you can submit up to 3 proposed formulations for your generic product. And please submit complete information about all excipients you plan in your generic product and the concentrations of excipients inside the container of your product. For example, if you are developing a generic metered-dose inhaler, the concentration of excipients inside the canister. With respect to device, the common question we receive is if a proposed test device is acceptable for an ANDA submission. As Dr. Witzman presented you earlier, currently we rely on the comparative analysis to access test devices in comparison to the reference devices. But the way we look into these comparisons is regarding to user interface. So when submitting device-related questions, submit samples of your proposed test product in addition to the reference product. Submit results of your comparative analysis, and based on the results, if you identify minor differences or more than minor differences or no differences, then you formulate your question. This will help us to with our assessment. This is just a snapshot of the guidance that Dr. Witzman already presented. Sometimes we receive questions if a proposed clinical study protocol is acceptable. So in this case, I want to make the point that we do not pre-review clinical protocols. And as Chris Andrea presented yesterday, you should be submitting specific questions regarding to your clinical development program. For example, if the PSG recommends a clinical study with formulation with population A, and you want to conduct your clinical study with a different patient population, this would be a type of question you should submit in your control correspondence. If you have multiple questions in your clinical development program, then we recommend you to submit a pre-ANDA meeting request. Sometimes we receive this type of question if the product is eligible for waiver of in vivo studies. In this case, I want to make a point that inhalation and nasal drug products, they are complex products. In this case, in vivo bioavailability or bioequivalence may not be self-evident. In that case, just waive the in vivo studies based on the CFR 32022 may not be appropriate. So if you are developing your generic product and you believe you don't need to conduct in vivo studies as per the recommendations in the PSG, you should be submitting an alternative bioequivalence approach in lieu of the clinical study that is currently recommended. In this case, in addition to your proposed approach, you should be submitting your rationale and justification. And if you have preliminary data to support your proposal, you can also submit that. And as final comment, many times we receive open and non-specific questions like, is my ANDA acceptable for review, or is the ANDA acceptable for filing? And then many times, if the ANDA can be approved. So at the pre-ANDA stage, we cannot address this type of questions because they need scientific review after the ANDA is submitted. So at the pre-ANDA stage, we recommend you ask specific questions about your development program, specific issues that you need help from the agency through the pre and the meeting request mechanism. So in conclusion, oral inhalation and nasal drug products are complex drug device combination products. We publish product-specific guidances to help development of these products. And the current recommendations in the guidances, they identify the current thinking methodology we believe is the most appropriate to support an application. If there are questions, clarifications that are needed regarding to the recommendations we have, or if you consider submitting an alternative bioequivalence proposal, you can do that. But please clearly identify your questions, focus on complex situations in your development program, provide your rationale and justification, and preliminary data if you have available. So I'd like to thank you, my co-workers, and thank you, audience, for the attention.